Hello, and welcome to part 2 of chapter 10, the cell cycle. In the first part, we talked about prokaryotic binary fission. We introduced eukaryotic chromosomes, introduced the cell cycle, and talked about the DNA replication process. So now we're ready to go through the different stages. So we talked about G1, where the cell is just carrying out its normal functions and growing to reach mature size. And then at some point it may get a signal telling it needs to divide. So then it goes into the S stage of interphase, where it makes a copy of the DNA. So that's the DNA replication we went over. After that's complete, it will go into G2, which is the prep for division. This is where you have the synthesis of proteins, microtubules will reorganize, and the centrioles will make copies of themselves. They will replicate. And of course, this happens only in animal cells because plant cells do not have centrioles. Chromosomes will begin to condense. That means they will pack themselves up. The copies that were made during the S stage will attach to each other at a location called the centromere. So that's what you see here in this image. So we have the centromere, which is the location where the two copies are attached. The two copies, by the way, are known as sister chromatids. So this is one sister chromatid, and here is the other sister chromatid, and they're attached together, and these are identical copies. Now, another structure you need to know about is the kinetochore. Kinetochore are proteins that are attached on the outside of the centromere, and their function is to attach the chromosome to microtubules, and we will see that here shortly. So once the cell has completed G2, it's all prepped for cell division. It will then enter mitosis, and in mitosis, it's divided into 4 or 5 stages. Your book divides it into 5, so we're going to go over those. The first stage is prophase. It's important to keep in mind that this is actually a continuous process. There's no distinct dividing line or end of one phase and beginning of another. It all happens quite continuously and smoothly. So as the end of G2 is happening and we've prepped for mitosis, we're beginning the first stage of mitosis, which is prophase. So the chromosomes will finish condensing. Here's our prophase image here. And a structure known as the spindle will begin to be assembled. Now the spindle is formed from the centrioles, and they will begin to migrate to opposite poles. So opposite ends of the cell are referred to as poles, just like you have a north pole and a south pole on our planet. So the centrioles are going to move to opposite poles, and while they're moving, microtubules begin to form and spread out from those centrioles to begin the formation of the structure called the spindle. The nuclear envelope surrounding the chromosomes is going to begin to break apart. It's being dismantled. Any nucleoli in the nucleus are also being dismantled as well. Now the spindle is the structure that's actually going to do the separating and moving of the chromosomes. The spindle consists of spindle fibers. These are microtubules that extend from pole to pole. In animal cells, you will also see structures made out of microtubules called asters. These microtubules extend from the centrioles out to the cell membrane in animal cells to anchor the spindle, because animal cells do not have a cell wall to keep all the structures inside in a particular orientation, which is needed for mitosis. The cell will begin to move into what we call Prometaphase, and some books don't have this stage. They just smush it in with the next phase. But this is Prometaphase. So let me see if my pen will actually work this time. Prometaphase. Okay, so Prometaphase. In this stage, the chromosomes become attached to spindle fibers. each sister chromatid is attached to opposite poles. So if you can see here, and I'll use my green color so it stands out, this chromosome, duplicated chromosome here, has one sister chromatid attached to one pole, and the other sister chromatid is attached to the other pole, and that's the same for the other 3 chromosomes in this image. The next stage of mitosis is metaphase. In metaphase, the spindle fibers align the chromosomes at the center of the cell. That is, the microtubules will move the chromosomes so that they line up in the center of the cell in an area known as the metaphase plate. This is a plane through the center of the cell where the chromosomes are lined up. Now, under the microscope, it looks like a straight line. It looks like they're kind of lined up. But in this bottom image here, you can see when it's tilted on its axis, so you're not looking directly at the equator of the cell, but off and an oblique angle, you can see that the chromosomes are actually lined up in a circular orientation. But they still are in the same plane. So if you took a ball and you sliced it in half and opened it up, that flat surface is a plate, and that's what we refer to as the metaphase plate, and that's where the chromosomes will be oriented in a circular fashion. They are lined up here to make it easier for the cell to separate them, which is the next stage of mitosis, anaphase. Okay, so this is anaphase. Yay, finally got something written. Okay, this is considered the shortest stage of the cell cycle. The centromeres holding the 2 sister chromatid copies together will separate, and the microtubules will pull the sister chromatids towards opposite poles. So you can see them being separated here in this image. Happens very quickly, and that's all there is to it. The last stage of mitosis is telophase. And here you have basically the opposite of prophase going on. So once the chromosomes or the chromatids reach the poles, they are now starter chromosomes. At least that's what they're referred to as. And the spindle apparatus will begin to be dismantled. A nuclear envelope will form around each set of chromosomes, and the chromosomes will begin to decondense. Nucleoli will reappear in each new nucleus, and you will end up with a cell with 2 new nuclei. And that's the end of mitosis. Now, because this is a very fluid process, it often overlaps with the next phase of the cell cycle, but doesn't have to. Even some cells will not go through this last phase that we're about to discuss. Let's discuss it first, and then we'll talk about the exceptions. So the last phase is cytokinesis. Cyto refers to the cell, kinesis is the movement, so cytokinesis is defined as the division of the cytoplasm. This divides the cell into 2 equal halves and forms 2 daughter cells. Now, it's done differently in animal cells than it's done in plant cells. In this image we have is 2 animal cells, and they go through cytokinesis using a process known as cleavage furrow. So the cells will constrict along the metaphase plate, or the equator of the cell, using actin filaments that are in a ring around that center location. And so they will begin to constrict and cause this furrow, and we refer to it as a cleavage furrow. And it'll eventually pinch the cell in 2. In this image on the right, the 2 cells are pulling apart from each other while they are pinching in. Plant cells do this process in a different way. They can't pinch in their cell membranes because their cell walls are rigid and won't go with the cell membrane, and you'll end up with 2 cells in the same cell wall, and that doesn't work for plants with their osmotic pressure. So each individual cell has to have its own cell wall. So the way plant cells do cytokinesis is by a process known as cell plate formation. cell plate formation. And I'm just going to abbreviate that. Okay, so here is an image of what's going on. We have an electron micrograph image on the top and a drawing of it on the bottom. So a cell plate forms in the middle of the cell along that metaphase plate. And the way it works is vesicles will pinch off from the Golgi bodies containing cell wall material. The vesicles will travel to this metaphase plate and they'll fuse together. And as they fuse together, the cell wall material inside will assemble into a cell wall, basically dividing the cell in half by building a wall. And the membranes that made up the vesicles will become the new plasma membrane on that side of the cell. So they'll fuse together as well. So you can see in the bottom image where the cell membranes are connecting with the original plasma membrane of the cell here and here. So eventually each cell will have its own cell membrane, and in the center we'll have cell wall material. It builds from the center, inside center of the cell outward till it meets the outside cell walls and cell membrane or plasma membrane, and effectively dividing the cell in two. So that's the end of part 2. Please watch part 3 where we'll talk about the controls for the cell cycle. Thanks for watching.